this is something in my 30 years experience, this really excites me. So if you keep your attention for the next few minutes, I'll share with you, see how and why I'm excited. So this slide illustrates one of the major problems in public health, that is the post-infarction airway remodeling and heart failure. Every year, we spend trillions of dollars in the world to take care of the post-infarction airway remodeling and heart failure. And replace this thin piece of scar with myocardium is the dream of our community. So remuscularization of the injured ventricle has been active research in my lab, and this remuscularization of injured ventricle has been successful in the past 2 years in the rodent model by using the human iPS cell-derived MHC-driven cyclin D2 overexpression myocyte transplantation into the immunodeficient mice. So this is 4 weeks after the transplantation and 6 months after the transplantation. The red indicate human-specific troponin T, a significant piece of a muscle in the rodent immunodeficient heart with injury at 4 weeks and 6 months. So by 6 months, with this human iPSC-derived MHC-driven cyclin D2 overexpression myocyte, the 50% of every scar is replaced by the muscle. So there can be achieved in the immunodeficient rodent, whether it can be achieved in large mammal. That's our focus in the past few years of study, and that is the our hub folks. So today, in the next 15 minutes, I'll talk about the myocardial remuscularization from within and outside in large mammals. Within, I mean to turn back the clock of cell cycle in neonates. And the second part, if time allows, I'll talk about transplantation of cells, cell products, and engineered functional myocardial tissue patch in heart with a post-infarction airway remodeling. This slide illustrates the Hashan CDEX report in about 10 years ago that in rodent, myocardial remuscularization can occur, but only occurred within the first 7 days. As shown here, the apex resection of postnatal day 1. By postnatal day 21, the apex is regenerated in the neonatal rodent. Whether it can happen, this significant remuscularization can happen in large mammal is significant because the difference between rodent and large mammal. So in late 2018, there are 2 reports published simultaneously in circulation demonstrating that the remuscularization of injured ventricle can occur in large mammal, namely in pig, but occurs only in postnatal day 1 or day 2. So reported from the UAB and separate in National Heart Institute from Singapore. So this can only occur in pigs in postnatal day 1 or day 2. So the significance of the remuscularization in large mammal is limited by the very early window. We reasoned if by 4 weeks apex is remuscularized, there must be active machinery of ongoing for the 4 weeks during the repair. So we want to examine if the activation of this machinery and during the 4 weeks, when we apply a second injury of AOD ligation, whether the heart can repair itself. This is important because this will expand the window from postnatal day 1, day 2 to 4 weeks. So protocol is illustrated here. This study is recently published in Circulation. So we have the study group of postnatal day 1 pig come in to do the apex resection, animal close the injury repaired, and chest closed in layers. By postnatal day 28, animal come to lab again. We did thoracotomy and ligate AOD distal to the second diagonal, and chest closing layers, animal repair, allowed to recover. By postnatal day 56, come back again, explant the heart to examine structural evidence of remuscularization. Subgroup of animals at 2 days and 7 days after the AOD ligation, we explant the heart, examine the evidence of early after AOD ligation if there's infarct by TTC staining. The control group 1 is the animal come in at postnatal day 28, have thoracotomy and ligate AAD, and explant heart at P56. And then we did another control group of postnatal day 1, day 28, day 56 to explant heart, examine the as a control group. We also have the single nuclear RNA-seq analysis of cardiac cell in neonatal pigs. This slide illustrates the typical postnatal day 56 heart with the heart with P1 apex resection, P28 AOD ligation. This slide illustrates the angiography of the before the explant heart at the P56 AOD ligation is successful, as evidenced by this arrow pointing the completely blocking of the AOD and the significant collateral already developed 4 weeks after AOD ligation as shown here. So we examined very carefully structural evidence of a myocardial infarct as shown here. We sectioned the heart according to the coronary anatomy. So the anterior wall, anterior pulmonary muscle, and septal are perfused by AAD, and lateral wall, posterior wall, and post-pulmonary muscle are perfused by LCX. So each heart sectioned and examined carefully by P56, 4 weeks after AAD ligation. There's no scar in this heart. And then we examined if the AOD ligation in this heart at early days, that namely 2 days and 7 days after AOD ligation in animals with double injury, these heart are again with P1 apex resection, P28 AOD ligation, and 2 days after AOD ligation, explant heart TTC staining showing the significant LV scar as we've seen before. And similarly, TTC staining of day 7, there's significant LV scar. So, but by day 28, 4 weeks after the AOD ligation, there's no scar by naked eye or by under the scope. Control group, typically apex, anterior apex and septal wall thin AV scar is shown here. I have done thousands of pigs. When you ligate AOD distal to the second diagonal, it always end up a big piece of apex anterior septal wall scar as shown here. So this is very remarkable and very reproducible by naked eye or under the scope. So, and I think it's very significant. This is the first time to show that we can expand the remuscularization window from day 1, day 2, of less than 7 days in rodent, we can expand this window to 4 weeks and complete remuscularize injured ventricle. So if we can expand the window by week 4, we may be able to turn back the clock to remuscularize injured ventricle in adult. So that's for the future. I will talk more about that later. So what is going on in this heart with double injury? We have evidence from the 2018 publication and 2020 publication in circulation that a significant remuscularization is contributed in part by the myocyte proliferation. As shown here, the Ki-67 staining, the compounded data showing on the right. That's the Ki-67 positive over the total new myocyte, as shown here. The normal heart, naive heart at P1, normal heart at P28, P56. The Ki-67 decreased from this level at the P1, about 5%, to almost 0 in normal heart. In remarkable contrast, this double injury at the P28, P56, this activation of machinery as evidenced by Ki-67 positive is remain high and even higher than the P1. Similarly, PH3 is also significantly higher as compared to the normal heart already decreased to 0. Likewise, the Aurora B, this is Aurora B, symmetric Aurora B positive, showing the cell kinesis. The normal heart decreased to 0, as shown here at P28 and P56, and the ones with double injury remains high. So single cell nuclear NR-seq indicate the double injury make the myocyte immature. So this is P1 myocyte, P56 myocyte. They are completely different in different location, and double injury make the myocyte immature. So I will have to finish here. So this is just indicate the pathway analysis review increased hippo YAP pathway, as shown here. The double injury have increased nuclear hippo YAP activity. So in conclusion, we have established a large mammal neonatal model with double LV injury and established for the first time to prolong the window of remuscularization of myocardial infarction to postnatal day 28. We generated a single nuclear transcriptome of cardiac cells from neonatal naive and injured pig heart. The regenerative heart maintained a population of P1-like cardiomyocyte, which continue to proliferate and continue to prolong the window to P28 of remuscularization of injured ventricle. The pathway analysis highlighted the activation of hippo-YAP signaling pathway in cardiomyocyte population that is responsible for the increased machinery of proliferation response. The ongoing study aiming at identifying the regulators and signaling pathways that govern the myocyte cell cycle in neonatal pig. And we're aiming at targeting these regulators in adult pig using the modified RNA, or mRNA now we call it, and new AAV9 gene therapy to remuscularize the heart with myocardial infarction and prevent LV dilatation and heart failure. I want to thank my collaborator and student staff as shown here. I want to thank you